Good evening to everyone. Hello. Good evening to everyone. So welcome to the today's class, especially the chapter 2, then the class 18. Today we are running on the class 18. Then the topic of today is the carbohydrate digestion in the ruminants. So this is one of the important topic as the ruminants are very important animals which are going to produce the milk at the same time meat to the community is concerned. Hence, it becomes one of the important chapter and many questions may be expected from this topic. And I request all the persons to join the class and hear so that I can also get some interest in explaining the material and also sometimes getting the things fooled or else it becomes very boring. Only one or two or three persons are joining, then that doesn't make any sense. It's okay anyway. So the ruminants, as the veterinarian, you know very well. I will not take much time on these things. It starts with the esophagus, then the rumen is a very big fur vat, or it is a very big 150 liters capacity thing is there. Then apart from this one, the fourth stomach is called as rumen, reticulum, omasum, and also the abomasum is actually real stomach. Four stomach are the three. Whereas apart from this one, the as usual regular, the intestine, small intestine, large intestine, ileum, rectum, and colon are also the part of the system of the digestive tract of the animals, especially the ruminant animals are concerned. Hence, we need to focus on the different parts as such. So, coming to the next thing, the ruminant carbohydrate digestion starts from the mouth, and in the mouth, the ruminants do have the salivary amylase, which digests the starch down into maltose, then plays a very small role in breakdown of the especially the food and which is in the mouth. And the ruminants do not have this enzyme, and not all monogastric animals secrete it in the saliva. Then the compartments of the ruminants, especially in the ruminant compartments, are, as you know very well, that is the rumen, reticulum, omasum, and also the abomasum. As you everyone knows, the rumen is a turkey towel-like thing, and reticulum is having honeycomb. Omasum is having multiple folds, whereas the abomasum is a true stomach or the glandular stomach, which is very much similar to that of the monogastric animal stomach. Hence, this is very similar to that of the non-ruminant stomach is concerned. Then rumen is the largest stomach compartment in the consists of several sacs. Means it is containing different types of the pores and different type of the mucous membranes. Then ruminants, major part of the microbial fermentation takes place in the rumen. It's a very big vat which is containing the saliva and also number of papillae which are of different size and shapes, which are main organ of the absorption, especially in the ruminants. In all ruminants, the papillae are most numerous and longest in cranial sac, followed by the caudodorsal blind that is there, and papillae follow provide most absorptive surface. So what about the ruminant carbohydrate digestion is dietary carbohydrates are primary for the plant and the fiber and sugars and also the starch. Then plant fibers are not digested by the hydrolytic digestion, but the microbial digestion, which is an important step in the ruminants, while sugars and starches are always digested by the especially sugars and starches are digested by hydrolytic digestion of the help of the mammalian. Different types of the enzyme system is present, and ingested carbohydrates are exposed to extensive pre-gastric fermentation is concerned, and most carbohydrates especially fermented by the ruminal microflora or the ruminal microbes, what you can say. And rumen fermentation is highly efficient considering the feedstuffs ingested. Different type of the feedstuffs are ingested, and some bypass starch may escape the small intestine. Almost all the carbohydrate is fermented in the intestine, but sometimes the bypass starch or easily digestible starch can bypass the rumen, and no salivary amylase but have the plenty of pancreatic amylase to digest the starch is present in the rumen, especially reticulorumen. It's also called as reticulorumen. Then ruminal contents are always containing a huge quantity of the bacteria, fungi, and protozoa. Then the fungi, as you know it very well, that this is the number of the fungi, especially 10 raised to 3 means 1000 per ml of ruminal liquor, the fungi are there and they are the least. Whereas the bacteria, they account to the 10 raised to 10 per ml of the rumen liquor. It is the contribution of the bacteria. Whereas the protozoa is number 2. They are present to the extent of 10 raised to 6 per ml of the rumen liquor. So these are all the ruminal microflora content. And apart from this one, some virus may also be there, but their role is not well understood. Then, apart from this one, the microbial populations, especially, they are digesting the aminolytic bacteria, especially starch, sugar digesters, and they are the fresh works because they utilize all these things for the production of the required material to the ruminant. So it can be called as the symbiosis or a joint life, or they have got the life in such a way that they can share it for the benefit of each other. Hence, it can be called as the symbiosis. So the rumen pH is, if it is 5 to 6, then there will be an appropriate bacterial growth will be there. Then they produce the volatile fatty acids. The They are called as the butyrates, then propionates, then sometimes the predominant animals fed grain and lactates also, propionate, butyrate and lactase. And apart from this one, enormous quantity of the methane gas and carbon dioxide are also produced as the byproducts of ruminal fermentation. The rapid change in the gain diet especially causes lactic acidosis. So it drastically reduces the pH to the extent of 3 or 4. Then the varieties of the unwanted bacteria may also grow. Then the rumen wall is damaged. The lactic acid is absorbed into the circulation and causes lot of degenerative and pathological changes, especially in case of the ruminal microflora population, which includes Streptococcus bovis, then the another bacteria which is many type of the bacteria is there. And here you can see this is a flow chart of the thing which is depicted by Dr. Rahul Dangi, who is a very good nutritionist, and from his website with his anticipated permission I have taken the slides. Then uh, these polymers, especially the celluloses, they cannot be digested by the monogastric animals. So they consist of the starch, cellulose, hemicellulose, and apart from this one, the ruminants also do have the lignins, which can never be digested by a monogastric animal. Hence, these pentose cycle and phosphoketolase pathway is there, which converts the all these things, especially cellulose, starch, and hemicellulose, to ultimately the VFAs in the form of the acetate, butyrate, and propyrate. And apart from this one, lot of methane and also the carbon dioxide are the end products. So coming to the first one, that's the starches. Starch are easily digestible carbohydrate available to any type of the animal, including the ruminants. So by the glycolysis process with several glycolytic enzyme, it is converted to hexoses. Hexoses. The starch is converted to hexose here. Then the hexose is converted to phosphophenyl pyruvate, a product especially in the ruminal content, especially microflora. Then this phosphopyruvate is converted to pyruvate. Then this is one of the ingredient that is very much required for several reactions in the body, biochemical reactions in the body. Then one carbon dioxide moiety is emitted out. Then this is converted to acetyl coenzyme A. Then this is converted to acetate, a volatile fatty acid. Later we will see how these volatile fatty acids contribute to the carbohydrate requirement of the animal. And once again, the cellulose also, with the help of the uh, bacterial microflora, it is converted to hexose, hexose to that another product called as the phospho pyruvate. Then the same pathway follows. So the pyruvate is also converted as lactate. Lactate is converted to propionate. Whereas the earlier product, phosphoenol pyruvate, is converted to oxaloacetate, acetate. Then oxaloacetate is converted to succinate, succinate to propionate. Once again, the VFAS. Then the pyruvate is one of the important end product of the glycolysis by several processes. For example, the celluloses, they are subjected to the microbial digestion, whereas the hemicelluloses are also converted to hexoses. Then, then apart from this one, the hexoses, then they are converted to pentoses, means 5 moleculed substance, or these pentoses are converted to glyceraldehyde 3 phosphate, then or the fructose 6 phosphate. Then ultimately this is converted to the hexoses. Then the pentoses are converted to acetyl phosphate, ultimately acetate. So this is another this process. Whereas the pentose cycle and the phosphoketolase pathway, wherein which the pentoses are broke down to ribose 5 phosphate, they are anabolized. It's called as the, instead of metabolism, they are anabolized to acetyl-CoA, propyl-CoA, and pyruvate and oxaloacetate. Then ultimately, the end product of the ruminant fermentation is enormous quantity of the acetate, butyrate, volatile fatty acids, and also the propionate. Then these are called as the VFS. Whereas the methanogenesis occurs with combination of carbon dioxide with the hydrogen atom, then the methane and the water molecules will be produced. Hence, we need to understand that this all is due to the microbial metabolism, microbial digestion, or microbial action. So how it occurs? The feed in the form of the starch, cellulose, hemicellulose, or sometimes lignin is being added with the microbial metabolism, wherein with the ADP is converted to ATP, and ATP is converted to ADP. Then biosynthesis of bacterial growth maintenance transport system will be maintained. At the same time, the NADP plus is converted to NADPH, then VFA, carbon dioxide, methane, and heat are produced. Usually, the rumen temperature is 1 degree always above than that of the animal's body temperature always, because the metabolism is such a fast thing and it happens always with that of the digestion process of different things. Especially we are dealing with the carbohydrate digestion, and apart from this carbohydrate digestion we have also got the digestion process of the proteins and also the fat digestion. We will see the tomorrow and day after tomorrow's class how they are all going on. Then here it's the another metabolic pathway of major carbohydrates of the plant origin to 3C, or it's also called as pyruvate units in the rumen. So this is the flow chart. Already it is explained. Once again, I will explain it by how it undergoes. So usually the uh, food of the animals do contain starch, cellulose, pectin, and hemicellulose. These are the four common ingredients, especially which are going to produce the carbohydrates. For example, the starch is converted to maltose, maltose is converted to glucose, glucose is converted to glucose 6 phosphate, and it is converted to fructose 6 phosphate. Fructose 6 phosphate is converted to 1,6 fructose diphosphate. Then this is converted to dihydroxyacetone phosphate. Then this is converted to glyceraldehyde 3 phosphate. Then 1,3-P glycerate. Then later 3-P glycerate is produced. Then 2-P glycerate is produced. Ultimately, as we have seen in the earlier chart, that the end before end product, that is the phosphoenol pyruvate. Phosphoenol pyruvate is produced, ultimately resulting in the production of the pyruvate. So this is the pathway how a simple sugar or the starch is converted to pyruvate, whereas the cellulose is converted to cellulose, then it is also converted to the glucose, of course. Sometimes the glucose is present only transiently, just thin. Then once again it will enter this pathway, glucose 1P, and all these things will lead to the pyruvate, and ultimately it is converted to the volatile fatty acids. Whereas the pectin is converted to pectic acid here. You can see, especially I am pointing out this thing and hope you can follow this. So pectin is converted to pectic acid, then this is converted to galacturonic acid, then galacturonic acid is converted to xylose and other pentoses. At the same time, the hemicellulose is converted to xylobiose, then it is converted to xylose, then the same type of the pathway, xylose 5P, then xylose 5P is converted to pseudo heptose, then fructose 6 phosphate. Once again, it is entering this particular starch pathway. That ultimately the pyruvate produced. So, end product of all these things, whatever it may be, it is going to be converted to pyruvate. Pyruvate. So, the pyruvates later they are all converted here as usual. The particular step, so it loses one carbon dioxide moiety, then acetyl-CoA is produced, then acetate is produced, butyrate is produced, then also the propionate is produced. So this is the way how a complicated polymers are being converted to the volatile fatty acids. Then volatile fatty acids are absorbed into circulation of the animal, then the required energy content will be produced. So then apart from this one, the pyruvate later what happens? Then pyruvate is converted to fumarate. Fumarate is converted to acetate. Then pyruvate is also with that of the thiamine, then it's converted to acetyl-CoA, then acetyl-CoA, then beta hydroxybutyryl-CoA, then the crotonyl CoA, then butyryl CoA, ultimately butyrates are produced. So butyrates. Whereas the pyruvates also converted to oxaloacetate, then maltose, malatate, then succinate, then succinyl CoA, then methyl malonyl CoA, ultimately propanyl CoA, then propionate. So here we have got two products. One is the butyrate and another one is the propionate. Whereas the lactose, if it is consumed by the animal, then it also converted to lactyl CoA, then acrylyl CoA, then propional CoA, ultimately propionate is being converted. So, in young ruminants, suppose a calf is there which has born today and it has to become a ruminant. But suddenly it will not become the ruminant, but it takes certain time. So, in young ruminants, the rumen and reticulum are not fully developed and are very small when compared to the adult ruminants, where in which this two-third part of the peritoneal cavity is occupied by the rumen, and it's about 150 kgs, and about 75 to 80 liters of the water it does contain, apart from that of the 25 liter of the saliva every time which is poured by different salivary glands. Whereas the reticular esophageal groove is a what you call as the tubular organ. It forms a tubular organ as the transitory movement, and it causes a fold of the tissue. Then, or the channels of the milk water which is sucked from the nipple directly goes to the omasum, to the abomasum, because through the ruminoreticular groove in case of calf. So this is a wonderful process in which the milk is not going to the rumen and not getting digested, as the calf do not have the well developed rumen. So this is a reflex mechanism. Whenever the calf starts uh, suckling, immediately the rumina reticular groove is going to be closed. Then it directly goes to the abomasum. The neonatal ruminant animal has no ruminal bacteria population, but from the birth, a newly born cow has not got any developed rumen, but later it starts developing the rumen, and sometimes the cultures like the bacterial cultures are also put from the mother and environment, particularly through the contact because of the saliva and also to the dung. Suppose a calf is having the close proximity with that of the cow, then there is a chance that it gets the microbial population through the saliva of a licking animal. Hence, the solid is then fermented by to the volatile fatty acids, which stimulates the growth and development of a rumen, particularly the growth of papillae for proper absorption. So this happens in several stages also. Then how the carbohydrate digestion takes place in the rumen is, as we have already explained the chart, usually the cellulose, hemicellulose, pectin, etc. are digested by ruminal microflora, and through the microbial fermentation in the rumen. So now you know that rumen is the biggest organ of the body, especially the gastrointestinal tract, which occupies almost two-thirds capacity of the peritoneal cavity. The purpose of rumen fermentation is to produce energy as ATP for the bacteria to use it protein synthesis and their own growth. and there are many uh, what we call as the hypotheses are there, how the protein is converted to the fat and fat is converted to the protein, and vice versa is also being happening in many of the domestic animals. So this is a wonderful process in which the most unwanted things of the fodder, feed, etc. are converted to the beautiful milk, then meat, and the dung and the urine, especially in case of the ruminant animals. So we need to have an appreciation to any such animals which produces a very good fat, also the very delicacious milk. The rumen wall is having very wide surface area, then it absorbs most of the VFS, and apart from this one, the acetic acid, propionic acid, then butyric acid, which are three volatile fatty acids. So here you can see the structure of the volatile fatty acids like acetic acid, C2, then propionic acid and the butyric acid. It's also called as the C4 here. You can see C4. So butyric acid is also little bit more complicated than that of the simple structure acetic acid. So the propionic acid is having more carbon moieties, and at the same time the three carbon moiety is there, then whereas the butyric acid is containing the four carbon moieties. The acetic acid only two, propionic acid three, 1 2 3, whereas the butyric acid it is containing the four carbon moieties. Then the proportion of the volatile fatty acids to that of the how much quantity is present in every animal is, please go through the particular chart which say that the predominant proportion of the VFA is that, especially acetic acid 65%, propionic acid which is noted by P is 20%, then butyric acid 9%, whereas the acetic acid is highest in most of the ruminants. Whereas the branched chain fatty acids are to the extent of the 5%, and N-valeric acid is to the extent of just 1%. So here you can see that the predominantly the hay diet is consisting, especially the if an animal is administered with very good hay of this particular proportion, then the animals can digest it very well. Then the total VFAs, maybe acetic acid, propionic acid, butyric acid, totally they become the part of 2 to 15 grams per liter. So a rumen liquor every day per liter it contains about the 2 to 15 grams per liter. Then the total weight of the acids produced by maybe 2 4 kg per day. All these volatile fatty acids 4 kgs per day. Whereas the propionic acid formed by primarily from decarboxylation of succinate when fibrous carbohydrates are fermented, especially fermented. Whereas high standard and sugar diet produces the lactic acid, or, or simply the simple carbohydrates. If it is administered to an animal, then it is going to convert it to lactic acid, and that's called as the lactic acidosis, one of the most dangerous conditions, especially in case of cattle. Coming to lactic acid, it is one of the strongest acid than volatile fatty acids, but absorb very slowly than that of the volatile fatty acids. So the lactic acid is main culprit for the excessive lactic acidosis whenever the animal is on the grain engorgement. You might have seen that the animals do suffer from the lactic acidosis. Go through the chart once again. It is the reputation. Pyruvate, of course, the end product of the all the three, especially the starch, the hemicellulose or the cellulose. So they always follow some common route, that is called as the pyruvate is converted to acetyl coenzyme A, which is one of the very important enzyme system. Whereas this acetyl phosphate, it produces the acetate. Whereas the pyruvate, once again, it is going to contribute to the acetyl coenzyme A. In simple word, what we call ACHA, acetyl coenzyme A. So the acetyl coenzyme A is produced to malonyl coenzyme A, then to acetoacetyl coenzyme A, then beta hydroxyryl coenzyme A, then to crotonyl coenzyme A. Then lastly it is converted to butyryl coester A, and it is converted to butyrates. This is one of the major ingredients of the feeds. Whereas the acetyl propionate is going to converted to